Greetings and welcome to the Biosafety Level 2, also known as BSL-2, training for DRI personnel module. This module is adapted from course materials originally developed by the EHS department at UCLA. We are grateful for their knowledge, expertise, and generosity. This module will start by presenting some background information on lab-acquired infections to help drive home why it's so important that you have this training plus laboratory-specific training on biosafety. We'll talk about risk assessment and what those numbers mean. We'll talk about the containment requirements in a Biosafety Level 2 lab and for Biosafety Level 2 work practices. We'll talk about emergency procedures, both in the event that you spill something, some of the cleanup procedures, and also some of the medical or other responses that might be required when things go wrong in a BSL-2 lab. And finally, there will be a quiz to test your knowledge. Let's start with some background information on laboratory-acquired infections. Surveys of laboratories in the United States revealed that between the years 1930 and 1978, there were 4,079 lab-acquired infections. Of those 4,079 infections, there were 168 deaths. In the next 20 years, leading up to 1998, there were 1,267 infections and 22 deaths. Now, in 80% of these laboratory-acquired infection cases, no accident or exposure was reported. In other words, the people who were affected either didn't know they got exposed, or maybe they did know, but they kept it quiet, they tried to clean it off themselves, and didn't file an official report of a lab incident or accident. More than likely, most of these people never actually knew they were exposed in the first place. So what these studies of lab-acquired infections tell us is that most of these mystery exposures where somebody never reports an incident or an unusual occurrence, but they still get a lab-acquired infection, it's assumed that aerosols are the cause of most of these mystery exposures. Thankfully, we also see that the number of incidents is decreasing, and since 1998, those incidents have further decreased. These decreases are most likely due to improvements in containment equipment, such as biological safety cabinets, engineering controls, and also better training. Before conducting any work in your laboratories that involves biological hazards, it's critically important to have a risk assessment performed in your lab. A risk assessment will focus on how to prevent exposures to personnel, how to prevent laboratory-acquired infections, particularly in the event that exposure does occur, and how to prevent environmental contamination, so those things you're working with don't contaminate other laboratories or other spaces at DRI. While conducting a risk assessment is officially the responsibility of the principal investigator, you can also receive assistance at this from Environmental Health and Safety. The Institutional Biosafety Committee at whatever collaborating institution you're working with, particularly if it's UNR or UNLV, can also be of assistance in conducting a risk assessment. Two very useful tools for risk assessment include knowing the agent hazards and what risk group an agent is in, and also evaluating procedure hazards. More details will follow. In laboratories where biological safety work is done, risk assessment is usually qualitative rather than quantitative. In other words, biosafety risk assessment is generally based on what you are working with, not how much of it you have. The particular pathogenic agent is of greater interest in preparation for safety procedures that may be in place than the particular titer or how many infectious particles there are per mil of that particular thing you're working with. Of course, contact DRI Environmental Health and Safety for assistance with risk assessment. All biohazardous agents that can be used for research at DRI are assigned to a risk group. In the United States, the National Institutes of Health is the organization that assigns risk groups to pathogens and other biohazardous agents. Risk groups will vary based on the agent hazards, with risk group 1 being agents that are not associated with disease in healthy human adults, And risk group 4 are associated with lethal human disease, for which there's no prevention or therapeutics readily available. Now, risk groups are not exactly the same thing as laboratory biosafety levels. You can have a risk group 2 agent, but you might require a biosafety level 3 work practices. While they're often similar, they're not always the same, so they both need to be looked up and researched. If you're wondering how agents are assigned to a risk group, the NIH uses 5 different criteria to determine which risk group an agent will go into. One is the infective dose. How many virions does it take, or how many bacteria, or how many particles does it take to create an infection? The environmental stability. Some agents are a lot more stable on fomites and other surfaces than others. The host range, whether the pathogen is endemic in nature, and also the routes of transmission and how easy transmission can occur. All of these things are used in order to classify an agent into a risk group. This table depicts the 4 risk groups as defined by NIH. Risk group 1 is not associated with disease in healthy adult humans. Some examples of pathogens that are in risk group 1 would be adeno-associated viruses and E. coli K12. Risk group 2 can be associated with human disease, but it's rarely serious, and there's often preventive or therapeutic interventions available if somebody does come down with them. Examples of this would include Salmonella and Klebsiella bacteria, along with adenovirus, hepatitis, and herpes simplex virus. Risk group 3 means that there could be serious or lethal human disease resulting from the agent, but there's generally preventive or therapeutic interventions available. Risk group 3 also means that you have high individual risk with an infection, but not necessarily a high community risk. Examples of this would be SARS-CoV-2, the virus that causes COVID-19, along with yellow fever virus, human immunodeficiency virus, human T-cell lymphoma virus, and the Mycobacterium tuberculosis bacteria. Risk group 4 is reserved for agents likely to cause serious or lethal human disease for which there are no preventive or therapeutic interventions available. They are considered both high individual risk and high community risk. And for this, we're mostly just talking about known hemorrhagic fever viruses such as Ebola, Marburg, and Lassa. We have to consider the routes of transmission if we want to prevent laboratory-acquired infections. About 1 out of every 5, or 20% of all lab-acquired infections, happen from either direct skin, eye, or mucosal membrane contact with the infectious agent, where a person incidentally inoculates themselves with the infectious agent from a contaminated sharp or from being scratched or bitten by an animal that's infected with the infectious agent, or from ingestion, where you get hand-to-mouth transmission. So if all of those are only 20% of lab-acquired infections, then that means that approximately 80% of all laboratory-acquired infections result from the inhalation of aerosols. Additional resources for conducting and understanding risk assessment are available at these URLs. These include the Biosafety and Microbiological and Biomedical Laboratories, 5th edition, the BMBL, which is sort of the Bible of biological safety here in the United States. There's NIH guidelines for research involving recombinant and synthetic nucleic acids. There's also useful information available at the American Biological Safety Association and also from the Government of Canada. So once the risk assessment has been conducted, at that point a biosafety level will be assigned to the work and to the lab hosting the work. And as a reminder, biosafety levels are often have the same number as the risk group for the pathogenic agent, but not always. Biosafety levels are assigned to both the lab itself and to the work practices that occur within the lab. And these are also not always the same. For example, if you are doing environmental specimen testing involving SARS-CoV-2, the agent which causes COVID-19, you have to have a biosafety level 2 laboratory for doing that work. But the actual work practices you will use in handling SARS-CoV-2 environmental specimens is BSL-3 work practices. So again, you're not always going to have the same number for the work practices, for the laboratory, and for the risk group. The purpose behind biosafety levels at both the laboratory level and the work practice level is similar to the purpose for assigning risk groups to pathogens. It's to assure that you have the proper procedures in place for containment so that biohazardous materials are confined and don't present a hazard to other researchers or the environment, and also to enhance worker safety and environmental protection. So the requirements of a biosafety lab, regardless of what BSL-2 it's assigned, is that it has to have standard lab practices and also special lab practices. There has to be containment equipment. You also have to take facility construction into consideration. These are all spelled out in the BMBL. Anytime an exposure incident occurs, be it a needle stick or an aerosol being generated outside of a biosafety cabinet, whatever the nature of the exposure, a thorough evaluation is critical for any incident that may result in exposure to infectious materials. Certainly, the incident must be immediately evaluated, and the person affected must be given treatment as quickly as possible. All forms of treatment that would be indicated based on the type of exposure. There must be procedures for how to respond to an incident in the laboratory's biological safety manual. And they have to be written down, they have to be explicit, people have to be trained on them. This is not a time for winging it or trying to guess what to do. And all incidents must be reported promptly to the lab supervisor so that further after-action items can occur. Medical evaluation, surveillance, and treatment are provided as appropriate, and written records must be maintained of everything that was done, every treatment that was used, and maybe some things to prevent the exposure from occurring again. If you're going to operate a BSL-2 laboratory at DRI, every BSL-2 lab must create their own laboratory-specific biosafety manual. The contents of this lab-specific biosafety manual must include the following: standard operating procedures for every procedure that involves working with the hazardous agent. The experimental designs need to be written and carefully followed. How to properly handle cultures and samples, the protocols for that. The hazards of the agents. This can often be just sort of copying the materials from the BMBL or from the NIH and making sure those are in the manual. Training records to ensure that everybody is receiving lab-specific training on at least an annual basis. Emergency procedures, again, like we talked about in the previous slide. What to do when things go wrong, making sure these are explicit and written and followed closely. Biosafety practices and equipment, and also biohazard waste disposal practices. Is this something that's going to have to be autoclaved, incinerated? You have to make sure that the waste disposal practices are carefully written in the biosafety manual and also followed. So the BSL-2 training that is required consists of both this current training module you're taking right now, but also laboratory-specific training. This lab-specific training must be conducted by the PI or lab supervisor, somebody with knowledge and expertise about what's going to happen in the lab. This training must be documented. This can be done by a sign-in sheet or any other format, as long as it's documented that the training occurred and when it occurred. This training is required at least annually. It may be required more often if there are any procedural or policy changes. You don't want to necessarily wait until 12 months after the last training if you've made some changes to procedures or policy. You want to get people trained on that immediately. So at least annually, but more often if there are any procedural or policy changes to BSL-2 practices. Access to BSL-2 labs must be limited or restricted. Everyone entering must be advised of potential hazards. This usually happens through a combination of training and also door postings. Everyone entering must be trained, again receiving both the general BSL-2 training being given right now and the lab-specific training, and they must meet entry requirements. For infectious agents that actually have immunizations available, these immunizations need to be done and that would be an entry requirement. Of course, many of the agents that are worked with do not have immunizations, but if there are immunizations available, they need to occur before somebody can enter and work in a BSL-2 lab. People may not enter the BSL-2 lab if they are at an increased risk of infection, and those infections may have serious consequences. Again, when we're talking about BSL-2 or higher agents, they're things that have the potential to infect humans and cause relatively serious illnesses. The doors leading into a BSL-2 lab will have the standard DRI lab door signage that we're all familiar with. However, it will also have additional signage. The additional signage required for a BSL-2 lab must contain at least the following. It must contain the biosafety level, in this case, 2. It has to have the biohazard logo on it that you see here to the right. It has to list the agents that are going to be used in the lab. If it's a lab where COVID-19 environmental samples are going to be handled, then SARS-CoV-2 would need to be on that sign. If it's a lab where Klebsiella is going to be handled, Klebsiella would need to be on that sign. The personal protective equipment required must be listed on this sign. Yes, even if it's already listed on the other standard DRI lab door sign, it also needs to be on this one. If applicable, the required immunizations for entering the BSL-2 space must also be on this sign. The contact information for the principal investigator and an alternate person must be on the signage. Yes, this is also going to be on the standard DRI lab door sign, but this is a regulatory requirement that it also is on this sign. If there are special entry or exit procedures, these also need to be on the sign. Work inside a BSL-2 lab requires some personal protective equipment that goes a little bit above and beyond the standard laboratory personal protective equipment. What's required in any BSL-2 lab is that you have to wear a lab coat, gown, or smock. You have to wear closed-toed shoes. You have to wear at least basic eye protection, such as safety glasses. You have to wear more advanced eye protection, such as goggles and or a face shield, when you have the potential to generate splashes or sprays, such as maybe you're opening Eppendorf tubes, and there's always the potential for a little bit of splash to occur when that happens. So for things like that, you would need advanced eye protection. Anytime you're going to be handling biohazardous materials outside of a containment device, such as the biological safety cabinet, you have to have the more advanced eye protection on. You also need that advanced eye protection if you're wearing contact lenses. It's also highly recommended that you wear long pants when you're doing any work in a BSL-2 lab, or really any lab at DRI. At the present time, there is no vertebrate animal research conducted at DRI, but respiratory protection would be required if you're ever going to be in a room with infected animals at a host institution where vertebrate animal research occurs, or if the risk assessment determines that respiratory protection is going to be required because there's a great potential for aerosolization and of aerosols, and that these might not be captured by existing engineering controls and containment devices. If you think that respiratory protection might be required, please contact Environmental Health and Safety because we would need to help with that risk assessment. We'd also need to ensure that medical evaluation, fit testing, and training is required. Respirators are different than other forms of PPE, and they have much higher requirements. All PPE must be removed before leaving the lab. If you have an anteroom that's connected to your BSL-2 lab, it's acceptable to take off the PPE in that room as well. Disposable PPE must be disposed properly, generally as red-bagged waste. It can be laundered by the institution as well if it's non-disposable types of personal protective equipment. Contact Environmental Health and Safety if you have any questions about the waste stream or the laundering stream of your personal protective equipment that's used in BSL-2 labs. Gloves must be worn in all BSL-2 spaces. The type of glove to be selected will be based on risk assessment. Again, you can consult with environmental health and safety if you have any questions. For most BSL-2 level work, disposable nitrile gloves are generally appropriate, but again, the selection has to be based on risk assessment. When appropriate, you might want to double glove. This does reduce tactility and is a little bit more awkward to handle things like pipettement, but still, where appropriate, it's good to double glove, if it's possible to do so. Gloves should be changed when they are contaminated or compromised, like when the webbing between the fingers breaks, then the glove needs to be changed. But also, a lot of times people don't realize that these gloves are not meant to be a permanent long-term barrier. If you actually get a droplet of infectious material on your glove, you need to go ahead and take it off and replace it more or less immediately. That's not really designed to be a barrier against something that's on it for hours on end. Do not wash or reuse gloves unless they're specifically like a heavy butyl rubber or other type that's designed to be reused. Remove gloves before leaving the lab. Disposable gloves should be treated as biohazardous waste and put in a red bag container. Make sure to wash your hands thoroughly after removing gloves because regardless of how careful we are, a lot of times we will contact the outside of the gloves with our ungloved hands whenever we're taking them off. Of course, the same rules that apply in all other DRI laboratories apply in BSL-2 facilities at DRI, which include there's to be no eating, drinking, or smoking within the laboratory spaces, no applying contact lenses or cosmetics, no mouth pipetting. Again, that should be fairly obvious at this point, but we'll reiterate it here. If you have a problem with pests, such as cockroaches or other types of unwanted critters in your laboratory, contact facilities. We have integrated pest management programs that we can use. You might have to shut down the laboratory for a day or two in order to do that, but yeah, don't let pest problems go unchecked. Contact facilities or environmental health and safety if you notice a pest problem in any of your laboratory spaces, including BSL-2 labs. It's just the fact that a lot of biological safety work involves syringes with needles on them. So there's some specific Sharps protocols to keep in mind. You have to use extreme caution to avoid auto-inoculation. In other words, sticking yourself with a needle that might have some of the infectious agent on it. But also aerosol generation. If you're not careful when you're using needles and other sharps, or even pipettes, you can sometimes generate little microaerosols. This is why it's so important to do this work always within a certified biological safety cabinet or other containment device. It's easy to generate aerosols when you're squirting things out of a syringe through a small hole. Try to avoid sharps if there can be alternatives that exist. I know sometimes there are not alternatives, but consider if there might be. When you are done with a sharp, put it directly into the sharps container. Do not recap it first. Put it directly in. Do not attempt to recap it. Needles must not be bent, sheared, recapped, or removed from the syringe before disposal. All of these things can put you at risk of accidentally auto-inoculating or sticking yourself, or also generating aerosols. So try not to do any of these things. Just dispose of the whole entire thing intact. In recent years, the Occupational Safety and Health Administration, or OSHA, has put a big emphasis on trying to get safer needle devices to be used, particularly in medical settings where needle stick injuries are one of the most common recordable injuries that happens in these settings. These images you see are from the OSHA Sharps e-tool. The URL for that is included here as well. And, as appropriate, these safer needle devices, which are used in medical settings, should also be used in research settings, again, if it's possible to do so. Some examples shown here are a syringe with a protective shield, a syringe with a retractable needle where after the injection is done, the needle automatically retracts into the body of the syringe, and also the blunt tip blood drawing needle that you see over on the right. Again, they don't completely take away the hazard of being stuck by an infectious sharp, but they can greatly reduce it. So if it's possible to use safer needle devices, if you need to use needles, they should strongly be considered. You can contact Environmental Health and Safety if you have questions about this. If you generate infectious sharps or broken glass, these have to be disposed properly. For sharps, you really need a puncture-resistant container that's conveniently located so you don't have to transport the sharp very far before you can get it into its puncture-resistant container. The ideal is to have a puncture-resistant, purpose-built container such as the ones you see here on the right, ones that have been specifically built and designed to receive infectious sharps. These will also tend to have the biohazard logo on them already, which is something you'd want to put on one of these containers if it contains infectious sharps. Ideally, you want to be doing this work inside the biosafety cabinet, and so therefore you also want to dispose of sharps within the biosafety cabinet. So having one that's big enough for your needs, but small enough to fit inside the biosafety cabinet is ideal. If you cannot do it in this ideal manner, please contact Environmental Health and Safety and we'll help come up with a solution for you. For things such as broken glass, where maybe you break a 10 ml pipetter or a flask or something like that, do not handle any broken glass directly. Use a broom and a dustpan, tongs or forceps in order to actually collect the broken glass. This glass, of course, needs to be put in a sturdy container such as a lined cardboard box, and if it's biohazardous, it needs to be appropriately labeled. Again, Environmental Health and Safety can help with the cleanup and the guidance of cleanup of these sort of things. Also consider whether you really need glass. For things such as evacuation flasks, it might be easier just to order and use plastic ones. These, if they're knocked over, will not shatter the way that a glass one will. So wherever possible, try to use plastic instead of glass containers, particularly inside a biosafety cabinet where you don't have a lot of space anyway and things getting knocked over is a foreseeable outcome. For the transport of infectious agents, or for environmental samples that might contain infectious agents, these potentially infectious materials must be placed in a durable and leak-proof container during initial collection and then the subsequent handling, processing, storage, or transport. The two examples you see here also have the biohazard sticker on them. It is strongly recommended that for all transport and storage of not just agents, but any kind of even environmental specimens that might contain hazardous agents, that you have these biohazardous logos on the container, along with other identifying materials about what the possible agent is, where it was collected, the normal things that would be on a container as well. Again, this rule is the same for all DRI laboratories, but especially for BSL-2 laboratories. Any animals and plants that are not research associated are not permitted within these laboratory spaces. So for all procedures with infectious agents that involve possible generation of aerosols, high concentrations, or large volumes, must be conducted inside of a biosafety cabinet, a certified biosafety cabinet that's been inspected within the past year. For example, let's say you're working with environmental specimens, and you have a large sample, and you want to pour that sample into many small Eppendorf tubes in order to spin down and try to separate a pellet from the supernatant. To actually aliquot that potentially infectious environmental sample into the Eppendorf tubes, you would need to do that inside the biosafety cabinet. You can spin the Eppendorf tubes down inside a centrifuge that's outside the biosafety cabinet, but then afterwards, if you're wanting to aliquot separate the pellet from the supernatant and open the Eppendorf tubes, that would need to be done inside the biosafety cabinet. Every time you open one of these tubes, usually a tiny droplet will fly out of the top. Those are the kind of aerosol generating procedures that must always be done inside a biological safety cabinet. So here are some additional examples of aerosol generating procedures that need to be conducted inside a biological safety cabinet. Anytime you're trying to plate cultures, using subculturing or streaking, holding an inoculating loop, this needs to be done inside a cabinet. Anytime that you flame the loop, in between streaks in order to sterilize it. Anytime you, after flaming a loop, before you continue streaking, if you want to cool it down, whenever you put it down in the culture medium, it kind of makes that little sizzling sound. There's little aerosols being generated when that happens. So all this needs to be done inside a cabinet. Anything involving pipetting. If you're mixing microbial suspensions, if there's any potential for a spill on a hard surface, this needs to be done inside the cabinet. And any manipulation with a needle or syringe, including expelling air out of a syringe, withdrawing a needle from a stopper, or injecting animals. Again, potential to generate aerosols needs to be done inside a biological safety cabinet. A question that's often brought up is, can you centrifuge potentially infectious materials outside of a biological safety cabinet? Some centrifuges are much too large to fit inside a biological safety cabinet, or they would take up a great deal of space inside the cabinet. You can centrifuge potentially infectious materials in the open laboratory outside of the cabinet as long as there are sealed rotor heads or centrifuge safety cups in use, so that there's no potential for aerosols to be generated during the centrifugation process. Of course, then, after the centrifugation is over, any further manipulations of the tubes needs to be done inside the biological safety cabinet. The facility and infrastructural requirements for a BSL-2 lab are laid out fairly explicitly in the CDC's Biosafety and Microbiological and Biomedical Laboratories, or BMBL manual. Environmental health and safety is also available to consult with ensuring that your BSL-2 lab is built to the proper specifications. But just keep the following in mind. These are the things that have to be in place within your BSL-2 lab at a minimum. It has to have self-closing, lockable doors. It needs to have entrance doors that open inward, not outward. There needs to be a sink near the lab exit, along with soap and paper towels, so that hand washing can be done quickly before exiting the lab space. While there's not an explicit requirement for eye wash station and safety shower within the lab itself, there does need to be one within 10 seconds of foot travel. So generally, that means in the hallway right outside, or in the anteroom or some attached room. There also needs to be special biosafety postings on the door, as was covered earlier. The labs must be created of sturdy casework that can readily be cleaned and decontaminated. This means that you can't have carpets or rugs or drapes or any kind of fabric materials, including chairs. You don't want to store cardboard inside a BSL-2 lab or styrofoam. It's strongly recommended that any BSL-2 lab facility does not have exterior windows. They also must have a negative or inward airflow, so that in the event that biological agents escaped the biological safety cabinet, they're going to stay within the room because there's negative inward pressure. The furniture inside a BSL-2 lab must be capable of supporting the equipment. Before putting a heavy rotored centrifuge on top of a piece of casework, you might want to consult with facilities to make sure that it's going to be able to hold up that load. This is not the place where you want to have things collapsing. The spaces between furniture must be accessible for cleaning. Bench tops must be impervious to water and resistant to heat, solvents, acids, alkalis, and other chemicals. And as said before, no cloth-covered chairs. Everything needs to be able to be readily wiped down and disinfected. All BSL-2 laboratories need to have a biological safety cabinet in them. These have to be Type 2, and they need to be certified on at least an annual basis. You also have to be careful where they're installed. They have to be located in such a place that room air supply and exhaust do not interfere with their normal operation. Where possible, they should be located away from doors, windows that can open, heavily trafficked areas, and other possible airflow disruptions. Once they're in place, and they're certified, then they're good, but some locations are slightly more ideal than others. If you're going to use aspiration flasks, For example, in order to remove supernatant from pellets, we recommend that these aspiration flasks be made of plastic rather than glass. But regardless of material, aspiration flasks need to be inside the biosafety cabinet, and the vacuum lines must be protected by a HEPA filter, which is replaced as needed. We do not want the vacuum lines to become contaminated with biological agents. Within the BSL-2 lab, all equipment and surfaces must be routinely decontaminated using an approved disinfectant, such as a 10% sodium hypochlorite bleach solution, or other ones as appropriate. You also need to decontaminate surfaces following a spill or splash. You don't want to necessarily wait unless you can't stop your procedure, you don't necessarily want to wait several minutes or hours after a spill before you start to clean it up. Decontamination must be done by staff who are trained to work with biohazards, and you should decontaminate equipment before repair, maintenance, or removal from lab. Certainly, if you're going to remove something from your BSL-2 lab, decontaminate it first, and then you might even want to put a posting on it saying decontaminated after it's done. You want to decontaminate all potentially infectious cultures and stocks before disposal. This can include cultures in tubes, in agar plates, wherever these cultures and stocks might reside. Autoclaving may be necessary in order to properly decontaminate these things, but you have to have a procedure in place and you need to follow it. You can consult with Environmental Health and Safety if you have any questions. And as stated before, for materials that are used for transfer of infectious cultures and stocks, such as sharps, make sure to use a proper hard-walled and labeled container for disposal of those. Over the next few slides, we're going to talk about how to clean up a biohazardous spill. Because, a lot of times this work is done in somewhat of a confined environment, and sometimes our hands will accidentally knock things over. Incidents will more than likely happen, so you need to be prepared for what to do in those cases. Now, you should only clean up a biohazardous spill if you feel comfortable doing so and if you feel prepared that you have the adequate supplies and training in order to do so. Your response to a biohazardous spill depends on the risk of infection and the location of the spill. Some spills are going to be more urgent than others because of these. The goals for cleaning up a biohazardous spill is to minimize the potential for infection to yourself, first of all, but then also the risk of infection to others, and the risk of environmental contamination from that infectious agent. The next goal is to minimize splashing and aerosolization. You want to clean up the spills in such a way that these are minimized because, obviously, that's not an exposure that we want you to have. Below are some general procedures that you might want to consider, though of course you should have laboratory-specific procedures too inside your laboratory-specific biological safety manual, so make sure to consult those as well. First of all, communicate to others in your laboratory that a spill has occurred. Call 911 if necessary, due to the injuries, the size of the spill, or if there's any other reason where you think this is a big enough emergency to necessitate outside emergency medical assistance. Make sure to wear personal protective equipment. You should be wearing this anyway when you're working with the agents, but absolutely make sure you have it on before you start trying to clean up a spill of this. The lab coat, gloves, goggles, and a mask. You want to place paper towels over and beyond the spill area. Don't just put them over where it is now. Put them where the spill might be headed. And then, once the paper towels are in place, you want to pour disinfectant on the paper towels and over the spill area. You want to do this very gently to minimize splashing. Once the disinfectant is poured, you want to let the disinfectant sit for the required contact time. If you're using a 10% bleach solution, there needs to be about 10 minutes of contact time before you start cleaning up the towels and the mess. If you're using 70% ethanol as your disinfectant, that requires a little more contact time. That takes 20 minutes. So again, don't get impatient. Let the disinfectant sit on the in the towels and on top of the spill for at least this required contact time. After the suitable contact time has passed, you can dispose of the paper towels, but they would be considered biohazardous waste. Then you want to clean the area again with the disinfectant and towels, again minimizing the splash while you do so. You want to dispose of any gloves that you used in the cleanup as biohazardous waste. Then after you take the gloves off, wash your hands very thoroughly with soap and water, and then report the spill to Environmental Health and Safety, along with your PI or lab supervisor. So if the spill does not just affect surfaces, but also affects you, meaning you get a significant amount of biohazardous agent on yourself, the first thing you should try to do is stay where you are. The only exception would be if you have a splash exposure to your eyes or face and need to go to the nearest eye wash or safety shower, in which case, yes, go to that. You want to remove contaminated clothing as quickly as possible, and then any areas of exposed skin, again, if you had to use an eye wash and safety shower, then of course you would do that, but if it's an area, just a small area of skin, like say on the arms or hands, you want to rinse the exposed skin with water, then wash it with a disinfecting soap, at least a couple of times. If necessary, seek medical attention. Under most circumstances, it is hoped that any spill that occurs will occur inside a biological safety cabinet because that's where all the potential aerosol generating procedures are being conducted. When this does occur, keep the biological safety cabinet fan running while you attempt to do the cleanup. Now, for really small spills of just a couple mils or less, you can finish working before cleaning it up. Again, if you're doing a time-sensitive assay of some sort, the thing with that, though, is you want to make sure to keep your sleeves and other items away from the spill area. And this is why we generally recommend that you go ahead and start doing the cleanup immediately after the spill occurs. But in those circumstances where you really can't do that, you have to be really mindful to keep sleeves and everything else away from the spill area before you start to clean it up. And then, once you start to clean up, use the disinfectants and towels, as mentioned earlier. There are situations where a splash, spill, or aerosolization will occur outside of a biological safety cabinet. For example, you could be transporting an environmental specimen or a culture tube to a biological safety cabinet, and it's sealed, but then you accidentally drop it on the floor, and it breaks open, and now it's not sealed. In the event that something like this happens, first thing you should do is evacuate the room and shut the door. Then you want to wash any exposed skin with soap and water. If you attempted to catch the tube or the specimen jar before it hit the ground and it didn't happen, and instead you got splashed, make sure to wash any exposed areas with skin and water. Do not re-enter the room for at least 30 minutes. The reason you want to wait is that this will allow any aerosols that were generated from the traumatic drop and splash and breakage to settle and vent. So by this time, if you wait 30 minutes before you re-enter the lab, most of these will have settled or vented out, and so there will not be any airborne aerosols anymore. It'll all be on the ground and on surfaces where it can be cleaned. So after the initial spill and exposure crisis has passed, if there's any non-emergency medical attention required, you should go to the nearest urgent care facility, depending on your location. If emergency medical attention is required, then the hope would be that at some point somebody would have called 911 and the emergency medical services would be on their way. Once the crisis has passed, make sure to contact the principal investigator and Environmental Health and Safety. We need to make sure that the incident report protocols are initiated. If the incident will result in lost work time, workers' compensation documentation protocols will also be initiated. So, in summary, Biosafety Level 2 work requires careful risk assessment, planning, and facility setup. You have to have all of them. You also have to have a laboratory-specific biosafety manual and lab-specific biosafety training. Above all, you need to make sure to wear your personal protective equipment, read and follow standard operating procedures, don't ever go off script when you're working with BSL-2 and higher agents, and be safe. And of course, environmental health and safety is available to assist in all of these planning and execution phases. So now you've reached the end of this training. All you have to do is complete the quiz and verification of training, and then you are finished. If you want further information, consult the DRI Environmental Health and Safety SharePoint site at the URL listed here. Then look specifically under the Biological Safety Programs and Documents page. This is where we'll post news, information, documents, and archives. And again, contact Environmental Health and Safety or your supervisor if you have questions or concerns. Be safe and well.